I also wanted to make sure that you guys understood some key aspects of the HIV life cycle. We are not going through this in the same level of detail that you would in a virology class. You can tell I just have my totally empty organelle-free host cell here, but I want to touch on the important aspects that we will particularly target in terms of drug delivery or drug design, okay? So here is my preferred host cell, a CD4 positive T cell, and here is my enveloped virus, HIV, okay? So the first thing that all viruses have to do in order to even start their life cycle is they have to be able to recognize their host cells. It turns out that for HIV, it has to have a receptor and then it also has to have a co-receptor, okay? So the preferred receptor here is CD4. And then for the co-receptors, there's two options. They can either recognize CCR5 or they can recognize CXCR4. So in order to properly recognize and bind to a host cell, it has to be able to bind to CD4 as well as one of these co-receptors. Okay? And so in the lecture, we talk about a mutation that essentially gives resistance to HIV infection. This is a mutation called CCR5 delta 32. There's a mutation in the CCR5 co-receptor that doesn't allow HIV to properly recognize cells with that co-receptor. So we would have receptor and co-receptor binding here by our virus. So binding is one thing. And then we would have viral fusion. So this is an enveloped virus, and the envelope from the virus is going to fuse with the envelope from the host cell. And so what this means is that for a period of time, that host cell will have some viral proteins on its surface. Those can be recognized by the immune system. The immune system can end up targeting and destroying these CD4 T cells. So that's one thing that leads to a reduction in CD4 positive T cells. The envelope will stay fused with the host cell, and then the nucleocapsid portion of the virus, so this is the genetic information, the single-stranded RNA, which is present in 2 copies, plus the protein coat, comes into the host cell. The single-stranded RNA gets uncoated, and so now it's free to be dealt with. So remember that what the virus carries is single-stranded RNA. Host cells don't have lots of single-stranded RNA, at least not stable single-stranded RNA. And so we have to find a way to convert this into something that host cells can use. And so the third stage is the use of the reverse transcriptase enzyme, which is a viral enzyme. Actually, if you look at the packaged virus as it enters into the host cell, it's carrying not only its genetic information, but also that reverse transcriptase enzyme with it. So this enzyme actually has 3 different types of activities, and it will allow it to go from single-stranded RNA to single-stranded DNA. You end up actually with an RNA-DNA hybrid right here, and then you can go from single-stranded DNA to double-stranded DNA. Now, double-stranded DNA is what our host cells have. And so at this point, what happens is you would actually get integration of that double-stranded viral DNA into our host cell genome. And so this is step 4. This is done by an enzyme called an integrase. That double-stranded viral DNA is integrated into the host cell genome. So again, another viral enzyme. At this point, this virus is now referred to as a provirus, and it is essentially sort of in a latent form. And so the virus can hang out here for long periods of time. It is invisible to the immune system at this point, and it's also not able to be targeted by drugs. So the virus can hide here for very long periods of time. This is one of the issues with actually truly clearing HIV from someone's body. Now, at some point later in time, this T cell will become activated by a normal immune response, something like antigen binding, and at that point, the provirus also becomes activated. And so the host cell will actually start to make messenger RNA from the double-stranded DNA that is incorporated into the host cell genome, and it's at this point that you get viral messenger RNA, and then that messenger RNA is translated. Now it is translated into these really long continuous proteins that are referred to as polypeptides or polyproteins. And so what you have here, you have all the viral proteins, but they're all connected together, and so they're non-functional. In order to actually make them functional, we have to have a viral protease enzyme that will come in here and actually cleave this long protein into functional proteins. And so once you have the functional proteins plus more viral RNA, single-stranded RNA being made, you can end up getting packages of the virus in terms of those nucleocapsids. You get that reverse transcriptase also, and then eventually these will be released from the host cell. The release step is another step that ends up leading to destruction of CD4 positive T cells. And so in terms of steps that we target with our drug design, we target co-receptor recognition and binding. That's a viral process. We target fusion of the viral envelope with the cell membrane. We target that viral reverse transcriptase enzyme. We target the viral integrase enzyme, and we target the viral protease. And so we have antiretroviral drugs in all of these classes that we can use to try to treat HIV.